Good morning, everyone. Welcome to Advancing Frontiers of Biotechnology, Exploring Gene Editing in Crops and Animals. This webinar is part of a series organized by ISA Inc. in collaboration with the DA Biotech Program Office. I am Janine Escasura from ISA Inc., and I will serve as your moderator for today's event. Recent advancements in biotechnology have been from the field of gene editing, genetic engineering, and gene drive. This webinar series intends to provide discussions on the new and emerging agri-biotechnology tools and innovations. The first of the series aims to provide information on the recent developments in the application of gene editing in crops and animals. As of now, we have more than 167 participants joining us today. Please be reminded that this webinar will run for 2 hours and will be recorded for documentation and evaluation purposes. So, before you start the webinar, please allow me to quickly remind you of the house rules. First, Zoom attendees are requested to keep their microphones on mute unless invited to speak. We highly encourage our audience to engage and actively participate during the Q&A session. If you have questions, you may post your questions using the Zoom Q&A button seen at the bottom of your screens. Please do not forget to indicate to whom you are directing your question. These questions will be addressed during the Q&A session. Lastly, please complete and evaluate the online evaluation form to receive your certificate. A link and QR code will be flashed at the end of the webinar, and only those who have accomplished the post-webinar survey will be given the e-certificates. Today, we have a set of activity objectives that we would like to achieve at the end of the webinar. First, this webinar intends to provide information about the concept and processes of gene editing and its various techniques such as CRISPR. Second, this webinar intends to provide an overview of gene editing applications in crops and animals. To formally start the webinar, we call on the Executive Director of ISA Inc., Dr. Rodora Romero Aldemita, to give the opening remarks. Dr. Aldemita is a multi-awarded scientist and science communicator. She holds a PhD in botany in Purdue University, USA, and a postdoctoral fellowship on golden rice at Albert Ludwig University, Germany. She coordinates the activities of ISA's Biotechnology Information Centers, as well as the capacity building activities on biotechnology and biosafety in Asia, in partnership with USDA and the Philippine DA Biotech Program Office. She is also a member of the Technical Advisory Team of the Applied Biotechnology Committee of the ABPO, a board member of the Biotechnology Coalition of the Philippines, the editor-in-chief of the Philippine Journal of Crop Science, and the scientific reviewer of 6 scientific journals. Everyone, please welcome Dr. Rodora Romero Aldemita.
Hello, good morning, everybody. I am very happy to be here in front of you, and welcome. Good day, good evening, everyone, wherever you are in this part of the world. And welcome to the ISA webinar entitled Advancing Frontiers of Biotechnology. And this is organized by the International Service for the Acquisition of Agri-Biotech Applications, or ISA Incorporated, with support from the Philippine Agriculture and Fisheries Biotechnology Program, or the ABPO. And at Erie, this is where our headquarters is, at Erie in the Philippines. So wherever you are in the world, we are broadcasting this webinar from our office at the International Rice Research Institute in the Philippines. This webinar is one of the ways by which ISA disseminates information about new biosciences and innovations, including genetic engineering, gene editing, genomics, gene drives, proteomics, among others. Different stakeholders should be kept abreast about recent developments in this biotechnology space. This webinar series intends to provide discussions on the new and emerging agri-biotech tools and products, which I mentioned. So this is the first of a series, and this webinar will be focusing on gene editing of crops and animals, because we know that gene editing is one of the innovations that is really going to drive food food production, environmental sustainability, and food sustainability as well. Currently, ISA has a lot of information in store at our ISA website. You can look at our gene editing repository at our website. We also have this gene editing of animals database, which will be launched during the virtual workshop, the ongoing virtual workshop on gene editing regulatory of animal biotechnology. And we're very proud to be a part of this virtual webinar, which is ongoing this week and next week. So this webinar is really very important and very significant because we have two STEM speakers coming from the academe, from the University of the Philippines, Los Baños, and the University of the Philippines, Visayas. Both of them are researchers and faculties or professors in their respective universities. So we would also like to invite everyone, since this is the first one, we will have a series. We'll have another one on gene drive and on another sciences, the emerging novel food. So with that, I'd like to reiterate the objectives that Janine has said. So we will talk about the process of gene editing and its various techniques, including CRISPR, TALENs, and zinc finger nucleotides. And this is going to be in crops and animals. And we're hoping that the science, the application on these crops and animals, the products in the pipeline will give you an idea of what is in store in the future. So with that, welcome once again to the webinar and have a great learning experience. Bye-bye.
Thank you very much, Ma'am Ola. Now, please allow me to introduce to you our first speaker for today. Dr. Maribel Zaporteza is an alumna and a faculty from the Institute of Biological Sciences at the University of the Philippines, Las Bañas. She has a master's degree in molecular biology and biotechnology and a doctoral degree in molecular biosciences and bioengineering from the University of Hawaii, Manoa, USA. She was trained with extensive molecular biology techniques at the Institute of Plant Breeding, which became her foundation to pursue other specializations in tissue culture, plant biochemistry, peptide engineering, mammalian cell culture, and neurobiology. After completing her PhD, she worked in the Greenwood Molecular Biology Facility at the Bekasi Neurobiology Laboratory at the University of Hawaii, Manoa. In 2016, she started working for the Philippine Genome Center for Agriculture program in the UPLB as a university researcher. To present gene editing in crops, please welcome Dr. Maribel Zaporteza.
Hello, can you hear me?
Yes, po.
Okay, so let's start. Let me share my screen. Okay, can you see my screen?
Yes, po.
Okay, thank you very much. Okay, let's start. Thank you for that very kind introduction. I would like to take this opportunity to thank ISA and the BPO for inviting me to share some knowledge that I hope you can learn something and at least the basics of the gene editing, I hope you can understand. And because this technology is going to be one of the very important technologies that would actually help in our food sustainability. So my assignment is to discuss gene editing in crops. So, okay, this is my presentation outline. So I will be defining gene editing or genome editing. And then I will introduce to you the most popular system that is used for gene editing, the CRISPR-Cas9 system. There are various other gene editing techniques, but we will focus, I will also mention some of them during this lecture, but the focus will be on the CRISPR-Cas9 system because this has become the most popular editing technology to date. Then I will also discuss a little bit of the differences between a GMO or a genetically modified plant versus a gene-edited one. So there, we should have a distinction on what is the advantages, let's say, of a gene-edited and the advantages of a genome modified. And then, of course, I will show some examples of the products of gene editing in plants. So what is gene or genome editing? Sometimes it's, for the purposes of this lecture, I'll just use gene editing because it's easier to say. But most researchers and some publications now really term it as genome editing. So genome or gene editing is a genetic mutation, or meaning there can be insertion, deletion, transversion, translocation of a predetermined genetic locus or a specific region in a genome using site-specific nucleases. So it's a long definition, but for me, the immediate term that I think about this is site-directed mutagenesis, meaning if you have a particular gene and you know its location, gene editing allows you to change the sequences of that gene in that exact location also. So there's actually two types of genome editing, the NHEJ, meaning non-homology and joining, and then the homology-directed repair. So in NHEJ, there is a double-stranded break that is introduced to the DNA. And then you can either have a deletion or insertion because the double-stranded break is going to be repaired, immediately repaired by your cell. Because if you have a broken DNA, that will cause problems. It should be intact. So your DNA will be repaired by your cell. And sometimes because this is an emergency, they will introduce some deletion or insertion. So in order to repair your DNA. And then the other technique is actually homology-directed repair. And this one, you actually introduce a specific template, and then you use the process of recombination to exchange fragments between your template and your gene of interest to introduce a certain sequence of DNA. So those are the two kinds of genome editing. So it is actually analogous when you change or edit sentences. When you write, let's say, when you write and you read an article, let's say the original sentence is "the girl is smart," and then you change the to "that girl is smart." So you actually change only a few letters of that sentence. So that can be considered as NHEJ. The next one, well, you can also use "she is smart." So if you look at it in DNA terms, you probably have shortened or lengthened the sequences of your gene. So that actually has implication later on for the mutation. It can either stop the transcription and translation of the gene, or either you can also alter the protein expression or the protein that's coded by the DNA. Another kind, you can use the same original sentence, "the girl is smart." You can replace "the girl" with "Anna is smart," or "she is intelligent." You now know the exact location of that sentence, and you know exact location of the replacement. So that can be analogous to homology-directed repair. And then you have the first generation genetically modified organisms, or the technique of genetic modification. So you actually can insert new characteristics, maybe coming from a different organism altogether. So let's say the girl is smart. Now you can change "the girl is beautiful and smart." So you now introduce a new character, let's say beautiful. Or now you can rearrange, let's say, the sentence as "the beautiful girl is smart." So that's actually first generation modification. So what are the enzymes that can act on this modification or changes in your DNA? And they are called site-specific nucleases. So these enzymes recognize unique sequence. And at these sequences, you can actually cut, make a double-stranded break. So it's actually a combination of a binding domain, meaning a part that can recognize the DNA sequence, and a cutting domain, which actually can break the DNA sequence in the two strands. So there are actually many kinds, there are actually four kinds of gene editing technique that has been used by researchers throughout maybe since 1980s. So the first one was meganucleases. So these are very much similar to restriction enzyme, except that they recognize bigger or maybe longer sequences of DNA. But because they are limited, but the sequence that they recognize, so we cannot use them for some specific locations in your chromosome or the DNA of your choice. So there's also zinc finger. So this is a little bit better than meganucleases. It's actually a zinc, it has a zinc cofactor, and it's made of a protein that can interact with your DNA. So here you can also have a recognition, but sometimes the recognition is limited because zinc finger acts as a dimer. So in order to introduce a double-stranded break, sometimes the zinc finger dimerizes or interacts with each other rather than your DNA target. So it's not, that's the limitation of this type of SSN. And then you have TALEN. TALEN is actually a very good genome editing tool. It's made out of units of, they call it tails. So it's made out of protein. Each tail or each unit can actually recognize a specific sequence of DNA. And in order to have a longer DNA that you can recognize, for let's say you want to design your recognition site or recognition binding domain, you have to design each of the unit of tail. So it takes quite a bit of expertise, but it is actually very specific and very good in genome editing. You will see later on that there are actually products that are made of tail using this technology. And the last one is actually was in 2013, this seminal paper that was published by Jennifer Doudna and Emmanuel Chapatier. They both won the Nobel Prize, is the CRISPR-Cas9 system, which is a little bit more advantageous than TALEN because now, instead of the protein unit that you design, now we design guide RNAs. And it's a little bit more affordable, and it's a little bit more, you can do a little bit more because you can now edit different sites within your genome simultaneously. I'll show you later. So all of them actually introduces the NHEJ or HDR. So we go to the CRISPR-Cas9 system. There's probably a lot more CRISPR system Cas9 Cas proteins now since the first publication of this paper. There are many kinds, but we will focus on the CRISPR-Cas9 because this is the classic one. So what is CRISPR? CRISPR is acronym for clustered regularly interspaced short palindromic repeats and the CRISPR-associated enzyme that can cut. So it's the CRISPR part is the recognition domain and the Cas9 is the cutting domain. And this CRISPR-Cas9 is actually part of the bacterial system, defense of the bacteria against the invading viruses. So the bacteria would actually recognize some sequences using this CRISPR system and cut the invading virus genetic material in order for it not to integrate in their genome and kill them. So they actually eliminate. So the cutting property is the one that the recognizing property and the cutting property is the one that they utilize in order to defend themselves. So it's actually a naturally occurring system in bacteria. So what is the advantage of this CRISPR-Cas9, CRISPR-Cas? Now we can now design the guide RNA rather than designing the protein, which is a little bit more difficult. And this creates several, you can also create several RNA guides that can be designed to edit a target gene at a faster rate and a more affordable cost. There was one question before, how affordable? Of course, it's a little bit pricey, but of course, let's say a milligram, I think 20 or 10 milligrams of protein is actually by maybe $100, depending on purity when you actually synthesize or order this. But for nucleic acid synthesis, you can actually order this now from some chemical company. You can write up to maybe $10 to $15, depending on the length. So here we see an example of a guide RNA. So in the top is actually what is found in nature. There is a CRRNA and a tracer RNA. Now this is now developed to be used in the laboratory, making it a little bit shorter and much more manageable, which is now the guide RNA. The one in pink is the one that we designed based on the DNA of your target. And the green one is actually, the green one is actually, sorry, I got confused with the color. It's actually the green one that you design. That's why it's N. It's based on the sequences of your target. And the pink one and the black one is actually part of the scaffold, which is recognized by your Cas protein in order to associate with the CRISPR guide. And then the Cas protein will be the one doing the cutting. So what are the requirements if you want to do gene editing in plants? So first of all, you have to have genome information. You have to know where the gene is located. What are your targets? Where is it located? Is there other copies of your gene in that genome? Next is you have to have a transformation process, meaning you have to transfer your guide RNA. How are you going to transfer your guide RNA to do the editing into your plant cell host? And then, of course, you don't want your plant cell to just remain as a plant cell, let's say a plate of tissues. You want them to grow into full-grown plants. So you have to have a regeneration protocol. And of course, you need to detect what sort of mutation have happened to your gene target. So you have to have an assay system. So these four, you have to have, these are the requirements of gene editing in plants. And one of the, so this is actually a diagram of how gene editing is done. So the green letters, that's N, that is your guide RNA. And one important component of designing your guide RNA will be the PAM or protospacer adjacent motif. In CRISPR-Cas9, which is the classic, the PAM is NGG. So you have to look for the NGG sequence in your gene of interest. And then design your guide RNA based on the other sequences next to the NGG. So here we have and then the Cas9 is actually behind. So if you find the NGG, actually the Cas9 will cut maybe 2 to 3 or even 4 bases away from before your PAM. So they will cut the double-stranded break, will be introduced 2 to 3 nucleotides before your PAM sequence. So here is an example. Let's say if you want to design guide RNA. So you have to look for sequences in your gene that has an NGG. N meaning it can be interchanged with A, C, D, G. But the GG is very, very important. So in this example, we found an AGG, and then you actually will start designing or get the sequences from your 20. So it can be up to 23 bases. You can actually get the bases and actually include this in here in your green, the green nucleotides here in the guide RNA design. So let's say here you have this, and then you copy this one, and you, of course, have actually, since this is just an example, this is just 50 nucleotides. Of course, a gene will be more than 50 nucleotides. So you can actually use different programs such as this. So these are free programs that maybe you are interested to try by yourself. If you have a specific gene of interest, you can try, and there's actually some the selection of the NGG is also very important, and also the sequences. There's actually some requirement. So that's actually when you design, when you read papers, and you need to rank your different guide RNAs that you will actually find from your sequences, and you can actually find the best one. So there's actually a criteria that you would have to follow in order to get the best guide RNAs that you can use for your experiment. So this is an example of a vector that you can introduce your guide RNAs into. This is a Cas9 binary and vector. So you see here, the guide RNA is located next to a specific promoter, and then you have the target region and the scaffold. So this is just an example. There's probably a lot more that you can find in published journals. So there's also you need to transfer your guide RNA or your vector containing the guide RNA into your plant cell host. And there's actually several methods that you can do that. But for plants, the most popular one will be biolistics and agrobacterium tumefaciens. And also some people also try to use tobacco mosaic virus. But the most popular one, at least at this time, will be the agrobacterium tumefaciens. So one of the critical points in plant gene editing will be tissue culture. This is quite critical. Not a lot of plants are amenable to tissue culture, and not all tissues are amenable for transformation. So this is actually a bottleneck. So in most laboratories, they now try to find the right cell type to transform, the one that can actually regenerate into full plants. Also, high efficiency of transformation. So transformation is also genotype dependent. Let's say in corn, some varieties of corn are very amenable to transformation, but tropical varieties are actually not so amenable. So you have to partner yourself if you want to go through this technique. You would have to really partner yourself with a good tissue culturist. So it's very important in plant genome editing. So this is just a workflow of the things that I've already discussed. You have to prepare and design your guide RNA, place it inside a vector, introduce it into your host cell, and then after the introduction, of course, you will select which of the plantlets will be positive for the transformation and insertion or editing, let's say, of your specific gene target. Then you will analyze it using sequence analysis to know what sort of mutation has been introduced. So what is the difference, let's say, of conventional gene breeding and genetic modification and gene editing? So here, what you see here is conventional breeding. Let's say our target characteristic is actually to get sweet taste and disease tolerance. So by conventional breeding, you cross two different varieties and screen its progeny, and then see if one of them actually has two of the characteristics that you want. So it might take some time and also some several breeding programs or several iterations, let's say, of a breeding program in order to get the specific variety or the characteristics that you might want. But let's say the characteristic that you want is really not found within the genome of your host, let's say in this case, tomato. So if that characteristic is not found in tomato, then you can actually isolate, that's gene modification, the first generation, is to isolate a gene coming from a completely different species and introduce it into your tomato. So that's actually the first generation modification. In gene editing, you actually have the gene is already there in the tomato. So there's actually susceptibility. So you want resistance and sweetness. So what we do with the susceptibility gene is to turn it off using editing, which means we can actually delete some parts of the susceptibility gene in order to turn it off. Now, since the susceptibility gene, or meaning something that is more prone for the tomatoes plants to get sick, is turned off. Now you get both of the characteristics, sweetness and disease tolerance. So it's actually the gene that you want to change is already there in the host cell. So this is actually a very simplistic, I think, diagram from BBC, that in gene editing, section of the DNA is actually changed, while in gene modification, a section of DNA is added, and sometimes it comes from a different species. So what are the examples of gene editing? So this is not a plant, it's a mushroom, but it's the first one that was able to get a green light from US government. It's the non-browning mushrooms of Pennsylvania State University. So these mushrooms were a big cash crop in Pennsylvania. And here they were able to turn off one of the 6 polyphenol genes that causes the mushrooms to get brown. And they were able to reduce the activity of the browning by 30%. And it was the first one because according to their screening, there was no detection of foreign DNA from virus or bacteria. So this is the first, at least in agriculture, something that you can eat. Not really necessarily plant, but as you can see, it's fungus. The next one is the first gene-edited plant that was released for consumers in the US is the oil called Calino by the company called Calyxt. So here, this soybean oil was found to be 80% higher in oleic acid and 20% less in saturated fats, and 0 grams of trans fat. And it has 3 times the frying life and longer shelf life. This product was developed using talents by introducing stack mutations in 2 genes of the fatty acid desaturase. So this is already available since 2018, I think. I mean, somebody can correct me with that. But this was the first plant that was actually approved in US. So next one is the gene-edited waxy corn. This is developed by a private company, the Dow DuPont or Pioneer Corteva. This one is designed to have all amylopectin in its starch, which is used very important in industry. And then the waxy gene, which is the gene that was edited, has been known for science for almost 100 years and has been used for conventional breeding. And this type of corn has been approved to be used in Japan as the 4th genome-edited food. So this is also approved in the Japanese regulatory process for genome-edited product, not necessarily for food, because the end product is actually amylopectin that is used in the food industry. So the first approved product in the Philippines is actually Tropics. That's the name of the company, non-browning gene-edited banana. So this company also developed their own gene editing technology, which is called gene editing inducing gene silencing. And this one was actually approved, recognized as non-GMO, and it is actually approved for importation and propagation in the Philippines. And there's another one is the GABA enriched tomato developed by Japan. This is developed in the University of Tsukuba. While the pandemic was happening, they were actually testing the tomato in the field, and now this tomato has 5 times the amount of gamma aminobutyric acid. It's very it's actually for heart health. So this is now commercialized in Japan, and this is the first gene edited food in Japan that was actually approved. And then this has this high GABA tomato is the second one that is approved here in the Philippines as non-GMO. So what does the future look like? Of course, there's in other countries and centers, research centers, there are various genome editing experiments and research. These are the crops that are being tested in various CGIR centers. So there's banana. Most of them are sources of carbohydrates. So banana, cassava, maize, potato, rice, and wheat. So these are samples of the specific gene targets that they would like to change and their characteristics that they would like to either turn off or increase. And this is just, it's actually a figure that I was able to get from Korotkova in 2016. So CRISPR was published in 2013. So this is in 5 years, they were able to, researchers now are targeting 81 genes in 16 crops in 5 years. So the progression or the use of this technology is really exploded. I think it's really a lot of laboratories are now looking at editing various crops that they are interested in, such as citrus fruits, of course, our staples, and of course some also some fibers such as cotton. So there are many researchers all over the world that are now using genome editing as a tool or CRISPR-Cas9 as a tool to actually modify or change some of the sequences of the DNA targets. So what is the take-home message for this lecture that I hope that you can actually remember? Genome editing is actually targeted site-directed mutagenesis. And the CRISPR-Cas9 system is the most popular due to its versatility and affordability. And CRISPR-Cas9 is actually a naturally occurring defense mechanism of bacteria against invading viruses. And then gene editing is also a promising tool to increase DNA variation, thus giving increased opportunity to develop advances in crops, animal breeding, and medicine. So I think that's it for my presentation. Thank you very much. And if there are, I think the questions will be after, right? Presentations.
Yes po.
Also, thank you very much. And I hope you learned something.
Thank you very much po, Dr. Zaperteza, for giving us an overview of the process and tools in gene editing. Everyone, we look forward to your questions. Send them through the Q&A function and we will answer them during the Q&A session. Again, thank you, Dr. Zaperteza. Let us give her a virtual round of applause. Moving on to our next speaker, Dr. Rhoda May C. Simora, an associate professor at the University of the Philippines, Visayas. Dr. Rhoda May Simora is also the OIC director of the Institute of Fish Processing Technology at UP Visayas. She graduated with a bachelor's degree in fisheries at UP Visayas and earned her master's degree in environmental biotechnology at Gyeongsang National University in South Korea. She obtained her doctorate's degree in fisheries and allied aquacultures at Auburn University, Alabama, USA. She recently completed her postdoc fellowship at Hokkaido University, Japan. She has worked on CRISPR-Cas9 gene editing technology and antimicrobial peptides as her research projects. She has authored and co-authored several papers related to CRISPR-Cas9 technology in Channel Catfish and has presented her work at both local and international conferences. Currently, she is the project leader of the Fish CRISPR project, funded by DA Biotech and implemented by UP Visayas in collaboration with the DA National Fisheries Research and Development Institute and DA Fisheries Biotechnology Center. To discuss gene editing applications in animals, please welcome Dr. Radha May C. Simora.
Thank you, Janine. Allow me to share my screen. Can you see my screen?
Yes po.
Okay. Thank you very much, Aiza, under the leadership of Dr. Aldimita for the invitation. My name is Rodame Simora. I'm from the University of the Philippines, Visayas, and I'm glad to talk to you, talk about gene editing in animals, particularly in fish, because that's my line of expertise. So my research experience, Okay, first slide. My research experience is primarily focused on CRISPR-Cas9 gene editing in channel catfish, and today I would be glad to share to you some of our results and published work on CRISPR-Cas9 gene editing. So the picture that you see on your screen, that is a microinjection setup on the far left of the catfish eggs. And then the middle picture, that's the catfish eggs with a capillary tube with the CRISPR-Cas9 components in phenol red dye. And then the picture on the right, that's the gene-edited catfish undergoing embryonic development. And the lower picture, that's the fingerling that has been gene edited. Okay, so the outline of my lecture will focus on three major subtopics. So I will discuss the overview of gene editing techniques in animals, particularly in fish, because I'm more familiar of gene editing in fish. And then I will also discuss recent developments in gene edited animals. And of course, the focus is on fish and the overview of the status of gene editing regulations in the Philippines. Okay, I know that Dr. Saporteza previously already provided you with substantial information about the basics of gene editing. So to provide you gene editing in a nutshell, this is the ability to make highly specific changes in the DNA sequence of the living organism. So actually, you're altering the sequence of the DNA in situ or on location at a specific point in the genome of an organism. It is done precisely and efficiently. CRISPR, Cas9, or other engineered nucleases is focused on precise and efficient gene editing. And the goal of gene editing is actually to leave no transgene footprint. But this is only true for gene knockouts. So when you say knockout, you abolish the function of a gene. But for another application, we call it gene knock-in, you're actually inserting an exogenous DNA to make a positive change, genetic change in an organism. Okay, so just to provide you a review of the gene editing technologies. So the common feature of the gene editing technologies, they have engineered nucleases. So when you say nucleases, these are enzymes that make cuts at specific DNA sequences. So Dr. Saporteza lectured about the four types of gene editing tools. So, but in fish, I assume that I know that meganucleases is not really used in fish before. So I will show you here the three major types of gene editing tools that were used in fish. So the ZFN, the zinc finger nucleases, the TALEN, and of course the CRISPR. So among the three gene editing technologies, so CRISPR is cheap, at least, and then easy to use and more efficient than TALEN and ZFN. And the advantage of CRISPR is that it is based on the natural immune system of bacteria, as compared to ZFN and TALEN, that they are synthetically produced and they are not based on the natural system. Okay, so to give you an idea of how this compare the three gene editing technologies, so based on these factors, so the CRISPR is actually based on an RNA. It's an RNA-based DNA recognition combined with Cas nucleases. So it is based on RNA DNA Watson Crick base pairing, and the CFN and TALEN, they almost have similar characteristics. The requirements for the ZFN and TALEN, you need to have 2 large protein constructs, but in CRISPR, you only need a simple 20 nucleotide change to the construct. And the targeting is good, feasibility is easy for CRISPR, and the cost is low as compared to the other gene editing tools. Okay, so these are the first applications of engineered nucleases in animals. So in 2009, CFN was first used in rats, and then after that in mouse and pig, and then rabbit and monkey. And then TALEN were also used in rats in 2011, and then pig, and then after that mouse, rabbit, and monkey. CRISPR was first used in 2013 in mouse, and then other animals followed like rats, rabbit, monkey, fish. Yung fish wala dito sa slide. So in fish, it was first applied in Atlantic salmon in 2014. I will show you later. So these are the major milestones in genetic engineering of livestock. So the first transgenic pig and sheep were made in 1985. And then after that, dito din, along this timeline, 1985 to 1990, the AccuAdvantage genetically enhanced salmon was made. And then after that, many animals were genetically engineered. And then in 2010, until the present day, ito na yung the advent of gene editing technologies. Okay, so this one, I'd like just to give you an overview of how a CRISPR Cas system work. So you only need 2 components. You need a guide RNA. So this is a specific RNA sequence that recognizes the target DNA region of interest and directs the Cas9 nuclease. So yung guide RNA is actually like a GPS. So ito yung nag-guide ng Cas9. Cas9 is a nuclease. It means that it's a molecular scissor. So para siyang molecular scissor that is this is the one that make a cut in a specific region in the genome to induce a double strand break. So that one was explained in detail by the previous lecture of Dr. Saporteza. And it can lead to several applications. So we have the SDN1, SDN2, and SDN3. So site-directed nuclease 1. So in SDN1, it will actually result to gene disruptions or insertions and deletions. So in this application, you don't need a template. You don't need an exogenous DNA. So the cell will, it's an error-prone repair mechanism that the cell will repair itself through non-homologous end joining. And then for SDN2 naman, after the double strand break, you need a homologous template that is homologous to the genome. So the gene modification is at 1 or more positions in the target genome. And then in SDN3, you need an exogenous DNA. For example, you need to insert an entire gene into a specific location in the genome. So this is appropriate for targeted gene replacement or DNA insertion. So we also call it gene knock-in. Okay, so those are the different SDN applications. And then in non-model fish or in commercial fish, CRISPR-Cas9 has been applied in some of this commercially important species. So we have Atlantic salmon, and then Nile tilapia, common carp, channel catfish. Most of the work and published papers on channel catfish were done by our team at Auburn University, and then sea bream and the rainbow trout. Okay. And these are the recent developments in gene-edited animals, or particularly in fish. So they introduced the term precision breeding. So precision breeding is different from conventional breeding and GMO breeding. So in conventional breeding, it would actually take many, many years for you to produce the desired trait that will be manifested in the phenotype of the animal. So kasi madami pa siyang mga crossbreeding na gagawin. And then in GMO breeding, the insertion is actually at random location. So you don't know where the gene will be inserted. So it will also take a lot of time and before you achieve the desired trait. And then in precision breeding naman, this is now the gene editing, because you are targeting the modification at that precise location. It will result to less time. So in animals, it will only take 1 or 2 generations. And then you will achieve your desired phenotype that will be manifested in the organism. So gene editing can actually forward the breeding process in fish from 30 years to 2 years. So in selective breeding, yung probability to inherit the desired trait is very low. When you do selective breeding in fish, it will take actually 30 years. And the probability that the next generation will inherit the desired trait is very low. And then in genome editing naman, it's very high, the probability to inherit the desired trait, because you make changes on the targeted gene on a specific location in the genome. So it will actually take 1 to 2 generations lang in fish. If your fish is fast growing like tilapia is can have sexual maturity in 6 months, it will take very short time lang, maybe 1 to 2 generations, less than 2 years for tilapia. And so why is fish excellent for genome editing? So fish have high fecundity, madami silang eggs na napoproduce. And then the protocols for artificial induction of spawning exist for many species. So you can actually let them, may time sila, pwedeng mag-schedule ka ng time to make them make more eggs. And then you can induce artificial spawning. And then the eggs are relatively large. So I showed to you the eggs of catfish. Very good, ideal siya for gene editing, yung catfish eggs, because it's transparent, the eggs. You can actually see where you injected your Cas9, CRISPR-Cas9 components, kasi transparent yung eggs. Fertilization is external and easily conducted in vitro. And then fish also have embryonic and larval development that occur outside the mother. So you can incubate your eggs, and you can also rear the larva and fingerlings outside of the mother. And then the generation time is very short. It's 1 to several years for many types of species. So mas madaling mag-sexually mature yung fish, mas madali siyang mas madaling ma-inherit yung trait. Okay, and do you know that in 2024 there are 3 genetically engineered food animal products that are commercially available in the US? So in 2015, I know you're familiar with the first animal that has been granted clearance for consumption, the first animal that has been pwede na siyang ma-consume publicly. It's the GM salmon, genetically modified salmon. It's a fast-growing line of Atlantic salmon that was made by Aqua Bounty Company. And then in 2020, there was the Gal-safe pig, which can be eaten by people who are allergic to a molecule called alpha-gal, na present siya dun sa unmodified pigs. And then in 2022, the FDA approved the gene-edited, short-haired, heat-tolerant beef cattle for human consumption. You can see on the picture, the two cattle, yung malaki, yun yung yung gene-edited. And the same sila na sister sila, siblings. So it has short hair than the wild type. The other one, the smaller one, is the wild type or the natural. So, mas malaki siya, short-haired, and this cattle can resist yung mga temperature changes, especially yung mga temperature increments during climate change. Is very ideal. So, very ideal ang cattle na to for climate change. So, it was approved in 2022. And then there are 3 gene-edited lines of fish that are advancing along the pathway towards commercial production. So, in 2018, there was the Nile tilapia, produced by the same company that made the GM salmon, but it was approved by Argentina and it was all cultured and produced in Argentina. And the modification is the myostatin gene knockout, which resulted to a 16% growth rate. And then in 2019, Japan, through the company Regional Fish Institute Limited, this is formed by 2 universities in Japan, the Kyoto and Kinda universities. They introduced the Red Sea bream, which is also a myostatin gene knockout, which resulted to a 20% more meat of the Red Sea bream. And then in 2021, the same company in Japan, they introduced the tiger puffer fish for commercial production and consumption. The modification is this is a knockout of the genes that control the appetite of the tiger puffer fish, which resulted to a 1.9 times heavier than the wild type. And then in 2023, December, last year, the same company in Japan introduced the gene-edited olive flounder, the fast-growing olive flounder, which was developed also by CRISPR-Cas9 gene editing. So yung olive flounder kasi, it's a very high-value fish in Japan because they use it for sashimi. Also in Korea. So when I was in Korea, I went to a fish farm with this olive flounder, and very siya talaga, very tinitake care talaga nila yung kanilang olive flounder because very precious for them. Because one plate, one fish, one dish of the sashimi of this olive flounder will cost around like 5,000 pesos. So ganun siya ka high value. And then how about the catfish that has more muscles? So look at the picture of the catfish na parang si Superman. Okay, that was our research at Auburn University Fish Genetics Laboratory. So yung premise, the research question is that what if we knock out the myostatin gene, the naturally occurring myostatin gene in catfish is similar to the cattle. Do you know that in cattle, they have this double-muscled phenotype cattle? In some breeds, the Belgian Blue and the Piedmontese cattle, they produce the double-muscled cattle, naturally occurring siya na mutation of the myostatin loci. So how about if we knock out the myostatin gene in catfish? Will it result to bigger catfish and more muscles? So, and the answer is yes. So we published a paper. So I showed the publication there, Nature Scientific Reports, that the cattle, the catfish that we knocked out the myostatin gene resulted to more muscle cells than the wild type. And then how about if we add alligator gene to reduce the infections in catfish? So the premise, the research question is that yung mga alligator kasi, they live in swamps in the US. They live in swamps and they engage in territorial fights and they have open wounds, but they recover. Hindi sila namamatay and they live for many, many years. And then scientists in 2016 published a paper that they have this cathepsilin gene in their blood that was shown to have strong activity against multiple gram-negative bacteria, but is not cytotoxic towards mammalian cells. And then my professor said, how about if we put in a cathelicidin gene into the catfish, and will it resist bacterial infections? And then the answer is also yes, because we also published our findings in that paper in Nature Scientific Reports, that we inserted the alligator cathelicidin gene into a non-coding region of the China catfish genome. And several publications after that were produced because we had an infection study. So yung mga fingerlings nung may alligator cathelicidin gene ng F1 generation na gene edited, we did an infection study with a bacteria, and then it proved that it's resistant to those bacteria. And then these are the pictures of full siblings of channel catfish produced at Auburn University. The top is the wild type, which is not modified, and then that is the bottom is the gene edited. So look at the size difference. And aside from the size difference that you can see, it's also resistant to bacterial diseases. How about healthier fish using CRISPR-Cas9? So this is the paper that we also published in 2022. So this one naman, we hit two birds in one stone. We did a knock-in kasi yung catfish kasi wala siyang masyadong, very low yung kanyang omega-3 fatty acid. If you compare it to salmon and sardines, so the goal is to make it have more fatty acid, omega-3 fatty acid. So what we did is we knock in a gene, we call it L of L2 gene, which has relations to the omega-3 fatty acid content. And at the same time, knock out, so abolish the function of the luteinizing hormone gene. So yung luteinizing LH gene, luteinizing hormone gene, is actually, the goal is to achieve sterilization so that if the catfish, for example, yung gene edited, goes in the wild, hindi siya makakamate. So that's the goal. So hitting two birds in one stone, knock in omega-3 fatty acid, at the same time, knock out the LH gene to make the catfish sterile. So that's the goal. Di ba ang saya? So two birds in one stone. So we call it multiplex gene editing. And then these are the successful applications of gene editing in some aquaculture species. So far, 18 species or more, counting pa, have been applied in fish. So with the use of CRISPR-Cas9. And the range of phenotypes are related to growth, reproduction, pigmentation, fatty acid profile, and diseases. So now for the status of gene editing regulations, actually may tatlo siya. For selectively bred fish, those that undergo the conventional breeding, there's no regulation. But for transgenic fish, the regulation is very lengthy process. So remember that the GM salmon, it took the Aquabounty company 2 decades, more than 2 decades, finally to get approval of the FDA. And for the gene-edited fish, meron dalawang regulation. So first is the categories pala. So there is the gene-edited fish without insertion or foreign DNA. You call it non-transgenic or transgen-free gene-edited fish. And the other one is the gene-edited fish with foreign DNA insertion. You call it transgenic gene-edited fish. Okay, so let's look at this global gene editing regulation tracker. Actually, you can track this on the website, So you just Google global gene editing regulation tracker. It shows the status of the regulations happening all over the world in each country. So meron siyang mga ratings guide. The green, dark green, it's determined, no unique regulation. They consider gene editing as the conventional siya na fish. And then mga red naman, it's prohibited. So like for example, the Europe part, they have limited, they prohibit gene editing. If you click Southeast Asia, it actually shows na information on, sabi sa information is countries in Southeast Asia have not determined the regulatory status of gene editing in animals. and there is little research or no products in development. So ganun ang status. And then yung Japan, it says here that it's lightly regulated. Gene-edited animals that do not contain DNA from another species are regulated as conventional animals because they already approved 3, the pufferfish, the sea bream, and the olive flounder. And they don't have tighter restrictions of transgenic GMOs. Yung may transgene naman na GMO, they regulate it lightly. So ganun. And then this is the status of gene editing regulations in the Philippines. We have regulations of modern biotechnology in the Philippines that is contained in the National Biosafety Framework in 2006, and then the Joint Department Circular number 1 in 2021, and the DA Memorandum Circular number 8, series of 2002. And for animals, we have the draft regulations for GM animals and animal products in 2022. And then I put in a box kasi it's still a draft guidance, and I'm part of the technical panel for this. The DOST is drafting the guidance document for the contained use of genetically modified and gene-edited fish, and also the draft guidance for the risk assessment of living modified fish for contained use. Hindi pa siya final, so draft pa lang. Okay, so this is the National Biosafety Framework. It is chaired by DOST, and those are, yung mga nasa orange, those are the agencies involved. And this is the agencies involved in the JDC1 series of 2021 circular. So ito yung mga agencies involved, the DOST, and the role of each agency, the DA, the DNR, DOH, and DILG. And this is an excerpt of the draft guidelines for the regulations of GM animals and animal products. So ito yun. That is particularly for animals and animal products. And then the gene editing Cas9, CRISPR-Cas9 gene editing efforts in the Philippines. We have established the CRISPR-Cas9 gene editing facility for fish at the Fisheries Biotech Center in Muñoz, Nueva Ecija, and UP Visayas. We inaugurated the facility on November 13 at FBC and December 7 at UPV. So FBC facility is designed to micro-inject. So we have the micro-injection setup, micro-injection, and then fish rearing, and then genotyping. And for the UPV setup, we have the bioinformatics and guide RNA design. So let me remind you for my last slides that the benefits of genetically enhanced organisms. So it will increase production, efficiency, and profits for the farmers, fish farmers, and it will lead to healthier, fitter animals, including the better animal welfare. And then genetically engineered animals are not organic, but if they reduce or eliminate chemical and antibiotic use, is that not as beneficial as organic? So we are advocating genetic improvement or genetic engineering or CRISPR-Cas9 as a solution to animal diseases rather than antibiotics or chemicals. And then thank you for your attention. I would like to show this picture. This is the fish genetics lab at Auburn University. The one on yung sa likod namin, those are experimental ponds for the transgenic fish that we had. And then we have a separate greenhouse for the gene-edited naman. And then this is yung other picture. On the right is the UPV and FBC Fish CRISPR team. And I would like to plug in 1 minute. I'd like to invite everyone. I will send an invitation through ISA. We will have a World Expert Lecture Series. This will happen in December 16 and 17, 2024. So you will see in person. So he will come to UP Visayas. If you wanted to see him, you can book your tickets and join us. So Dr. Rex Danham, Auburn University, my Auburn University professor, he is designated as being the top 1.5 scientist worldwide. He pioneered the research on catfish, CRISPR-Cas9 gene editing. He will have 2 public lectures. applications of CRISPR-Cas9 gene editing technology for commercially important fish species. And he will have another lecture on the 17th. It's in December pa, but I'm plugging now. And then lecture number 2 is transgenic and gene-edited fish. Is it safe to eat? So please join us. It will be a face-to-face lecture and online. So we will send invitation through ISA. Thank you very much, and I hope you learned something from the lecture.
Thank you very much po, Dr. Roda May Simora, for that very informative presentation. Definitely, the presentations of our speakers has given us more knowledge about gene editing and its applications in crops and animals. Again, thank you, Dr. Simora. Everyone, let us give her a virtual round of applause. So that concludes our presentation for today. We encourage everyone to post your questions at the Zoom Q&A function. For our Q&A session, we have Mr. Panfilo de Guzman, Senior Associate Scientist at SciSA Inc., to moderate the discussion. Sir Poppen, the virtual floor is now yours.
Okay, thank you. Janine, actually we received a lot of questions in the Q&A box. And the first question we have here is, what are the current advancement in reducing off-target effects in CRISPR-Cas9? And I think this is one of the possible problem areas when you do gene editing. And how do these approaches compare in terms of efficiency and safety in reducing off-target effects? Maybe we can have first Dr. Saportesa, and then Dr. Simora can add. Dr. Saportesa, please.
Yes. One way to reduce the off targets is to really have good genome, the genome of your target. You have to have the knowledge of the sequences of the whole genome. If you have the whole genome of your target, then it's easier to actually use the programs to avoid the off target. So some plants have already published their genomes, especially for the staple crops. That's why they are actually going ahead rather than the other crops that are targeted for genome editing. So there are various programs that we use, and of course the algorithm gets better and better with these programs. And then eventually also, even if with the program, it's still different when we apply it in the laboratory. So we still have to actually test them. We have a selection of guides, and then we have a test or the assay in the end of each experiment to see if there are any off targets. So I think it's really dependent on the host and the information that you have. It's really on the design of your guide RNA. So of course we actually can test. That's the beauty, I think, of the CRISPR-Cas system, is because you can test several all at the same time. So you can actually choose from them which one was actually the best. So yun yung advantage kasi niya. And it's a little bit more affordable than talent. So actually you can design several transformation experiments using different guides, and then you can see actually which one of the guides is actually giving you the better off-target, on target result na wala siyang off-target na natatamaan, or there's other genes that are getting also edited by your experiments or your system. So I guess we can also have, because she has more on-hand knowledge of Dr. Simora with
Yeah,
fish
Dr. Simora,
especially.
please. Yeah, if you can add some.
Yeah, I agree with Dr. Saporteza about the methods to assess the off targets. We have prediction software models, but the important question here is, how do we reduce the off target? So dapat ma-reduce natin by optimizing your CRISPR-Cas9 experiment. And how do you optimize CRISPR-Cas9 experiment? You choose the right guide RNA. And then second, you need to pick the correct donor DNA template if you wanted to do knock-in. And then third, you need to optimize your experimental conditions. So doon talaga yung pinaka ano. So you reduce your off targets. So yun po.
So you are saying that there are ways that we will not have any off targets, or
Yes, or
we
we
can
minimize. Yeah, we can
minimize it.
minimize. Doon pa lang sa
Okay.
experimental stage. Yes.
Okay, thank you very much. So there's a question here from an anonymous attendee, and he or she is asking, when going through gene editing and making a newer crop or altered traits, do you use it in seeds in the case of crops, or do you use it in eggs in the case of fish? We can have Dr. Saportes first.
Yeah. For plants, we usually, it depends actually on the kind of plant that you're using. That's why I was emphasizing a little bit on tissue culture. Some plants, like tomatoes, you can actually transform and target leaves, and then you get a whole plant using the tissue culture process. You have little plantlets of little tomatoes. But for, let's say, crops such as rice and corn, we transform the embryos that comes from the seed. So what we do is we excise the embryo out of the seed, a young seed, or let's say a kernel of corn. We will extract the embryo first before we transform them. Or in rice, you also extract the embryo from rice. So it's a little bit different from animals, but in plants, we have to get, that's why it's a little bit tricky in plants, is to get the right tissue to transform your gene editing vectors. So talagang medyo we have to choose which one will be the most amenable, highly efficient tissues that can generate the number of edited plants that we can screen for, let's say, a particular trait that we want. So for mom, a little bit different. Yeah.
Yeah.
Yeah, Dr. Simora, yeah, how it
Yeah.
is being applied in when it comes to modifying or gene editing the fish.
Yeah, I actually just learned now that it's how it's been done in plants. Thank you, Dr. Saportesa, for the information. For fish naman, I already showed you in the slide the microinjection setup. So we do it on eggs. Okay, so we put the CRISPR-Cas9 components in a capillary tube and we microinject it on the eggs. So ganon. It can be a cloning-free method, yung pag-generate ng guide RNA and the CRISPR-Cas9, or it can be using a plasmid. So ganon ang parang gene delivery method namin sa fish, sa eggs. So,
Okay.
yeah.
This question is addressed to Dr. Simora. Currently, we have the problem of African swine flu. Are there any research initiatives using the gene editing technology in addressing the problem of or producing a pig that is resistant to ASF?
I'm actually not familiar with the livestock because I work in fish.
Okay.
So, yeah. So
But have you heard of any initiative?
I cannot answer
Maybe.
the question. Yeah. But in terms of combating bacterial infections in fish, there are initiatives like, for example, you can do a gene knockout like what we did, or a knock-in. So ganun, parang mga ganun ang initiative sa fish. Like for example, that alligator gene that is resistant to bacterial infection. So we put it into the catfish so that the catfish will be resistant to bacterial infection. So parang ganun din siguro ang premise dun sa livestock.
Okay. And there's also a question addressed to you. Are there potential from James Ron J. Embargo? Are there potential long-term negative impacts of CRISPR-Cas9 on biodiversity and ecosystem, since this engineering has been used in agricultural practices na? So
Okay.
do you see any potential long-term negative impacts on biodiversity and ecosystem?
I'm biased. Maybe I'm biased because I'm a biotechnologist. So, but
Yeah.
on the report on the GM salmon, wala naman sila nakita na risk on the environment and biodiversity. But on my own experience with the fish, the transgenic fish and the gene-edited fish that we rear at the Auburn University lab, we do a lot of genetic confinement, physical confinement, lahat ng klase ng confinement, so that the transgenic escapes and the gene-edited na hindi siya makaka-escape sa environment. We're very careful of that. Very strict ang US sa ganun na mga risk na yeah. So I think we see to it. I think Dr. Zaportesa will agree that before we do an experiment, we see to it na the risk would be really minimal or walang risk dun sa biodiversity and environment. Ganun
Okay.
po.
I think there's a question now addressed to both of you. Can you use genetic engineering and gene editing techniques in one organism? So, meaning applying both gene modification and gene editing in one organism.
Yes.
Dr. Saportesa, please.
Yes, you can do
Then we-
both. Hello.
Yeah.
Yes, you can do both at the same time, which is actually makes it yun nga, multiplex na siya. So you can actually do both because in CRISPR, yung ang homology directed repair, that's actually considered knocking in. That's actually genetic modification. So it's not editing, it's also editing, but it's specific location. So you can actually do both using the CRISPR-Cas as long as you have the homologous template. You can design the homologous template. So it's actually the genetic modification before that was random ang insertion because of biolistics or agrobacterium or kahit microinjection. We did not know where the modification will happen in the genome of, let's say, a host organism. It can be random insertion. So this time now, because of the pairing of the next generation sequencing and this technology is really parang pair made in heaven, because now we know exactly where we want to insert. We can design the edit and analyze it using the same technique. If you have an edited plant or an edited animal, in my case plant, you can again resequence the DNA or the genome of the edited plant to know exactly where the edit happened. So you can also track where the additional characteristic is also placed. So para siya talagang is a pair. So you can do both, and it's really up to you which technique you can use and which is available for you.
Dr. Simora, do you have anything to add po?
Yeah. That's what I shared in the lecture, that we actually did a knock-in, simultaneously knock in a gene and knock out also a gene in the channel catfish. Yung we knock in yung omega-3 fatty acid gene, and then we knock out the luteinizing hormone gene that will achieve the sterilization in catfish. So we do it only one time in one injection. So yeah, as Dr. Saportesa said, we can do anything. So multiplex, we call it multiplex gene editing na.
Doon po sa presentation niyo, Dr. Sumora, you touch a bit on the draft regulatory guidelines for animal biotechnology. What particular government agency do you see na will be the focal point for the safety assessment of gene-edited animals or fish?
The agency that is concerned with the contained use for research, we are talking about research pa lang, not for commercial. It's the DOST, and then for the environment naman dun sa, it's the DNR and the DA Biotech, DA, yeah.
Okay.
So those are the focal government agencies.
How soon can you see that this regulation will be out?
We are drafting the the guidance document on specifically on GM animals and gene-edited fish. And I hope that they will finalize it by the end of this year. And then for the animal and animal products, it has been drafted. I'm not part of that team, but I saw the circular. It has, I don't know which step are they now, but it has been drafted in 2022. So I hope that it will get approved by the end of this year also.
Oh, okay. Thank you. Another question from our anonymous attendee. For the Philippines context, given the regulatory system and consumers' acceptance, is it really good to invest in researches involving gene editing of crops and animals? The output of current gene editing projects in our country might only fall into shelves. Dr. Soportesa, your insight on that one? Is it really
I
good to
think it
invest
is.
in this
Yes,
kind
it
of
is good to
researches?
invest, but we have to choose which ones we invest in because we're still in a developing country, so we have to really choose which ones we can invest in. So I think at least in the plant terms, in crops, we already have the regulations in place. So we just need a little bit more capacity building, I think, and the facility development. And we actually have the Crop Biotechnology Center already at PhilRice and several institutes. We also have ERI in the Philippines. So actually, ERI has several projects with gene editing already for rice. And for other areas, it's because we have to actually have the facility and, of course, the mode of capacity building also for our scientists, because it's a little bit different when it comes to crops. You remember that plant cells would always have some hindrance with regards to delivery because we have the cell wall, and that actually creates a little bit more of optimization to actually happen before we actually. But when we get the optimized protocols already, then I guess it would be just one crop after the other. So of course, it's long-term investment. It's not just now. Sometimes we think about research that, oh, you really need to have a product in the end. It's actually a long-term investment because products coming from other countries which are more advanced to us will be definitely coming in. So we have to have regulations in place, and we also have to tailor the plants or the crops that are important to us because they are not going to be done by other countries, because crops that are important to the Philippines are we are going to have to invest in them. So, I think it's worth it. It's just that we have to really select which ones to fund.
Yeah, Dr. Simora, what are your insights on this one?
Yeah, there are still legal and ethical uncertainties in gene editing, but it's here to stay. We cannot. Yeah, gene editing is here to stay. It has been it has revolutionized the field of molecular biology. We cannot do away with it. So we need to embrace it. So yung ano ko lang about the Philippine research on gene editing in fish or animals is that we need to focus on commercially important na mga livestock and fish. So we need to put our research yung focus there, and the support of the government talaga. In plants, medyo established na sila, but in livestock and fish, madami pang gagawin. So we need to do more. And the support of the government talaga in terms of funding, research funding, and approval. Yes, thank you.
There's a question for you, Dr. Sumara, here. Which stage in gene editing procedure is the most crucial? Are there visible indicators in the earlier stages in which you can already assess whether or not the editing is successful?
Yes, the most crucial when I talk about dun sa fish is that there is the microinjection step. You need to microinject the CRISPR-Cas9 components within the one cell stage. So to prevent mosaics mosaic phenotype. So dapat mabilis ang microinjection. And then before the cell divides, you should have injected the CRISPR-Cas9 component. So dun sa fish. So that's why you need to optimize. I said a while ago that you need to optimize your experimental conditions. So
Okay. Thank you. Yeah, for plant editing and gene editing, what could be the
Most crucial.
crucial stage? Yeah,
Yeah,
or
it's
the
actually the same. It's the
problematic area.
delivery. Yeah. It's actually the same stage. It's the
Okay.
delivery. But there's an additional stage for plants.
Mhm.
It's the regeneration. You can edit the cell, but it should be in plants, it should regenerate into a full-grown plant, or at least a plantlet stage, and it should not have any type of other mutation, because in tissue culture itself, you introduce somaclonal variations. So you have to avoid that. So you actually have to choose the right age of the cell that you are going to transform. And you also have to have an optimized regeneration protocol that in the media that your cells, or let's say an embryo that you have transformed, you have delivered the gene editing vectors or vectorless ones, it should grow, and there's actually steps. It should grow shoots first before the roots. If you have roots first, then you are in trouble. So it's a little bit more difficult than animal. I think it's the same level, probably a difficulty, because I have very bad hand-to-eye coordination. I probably would kill all the eggs. So, but in plants, it's like that. You have to have, that's crucial. That's why it's you have to be very observant, and you have to optimize that part of the experiment, because that's really one of the bottlenecks for both animals and plants. If you do not have the technique, the skill, so we really need people who are skilled and patient enough to do it, because it can probably take several, maybe, tries to really get the hang of transforming both plants and animals.
Okay, thank you. We have one participant raising his hand. John Ray Oronan. Yeah, you can unmute and you can ask your question, or you, John Ray. Can we unmute John Ray, please? You can start talking now because you are permitted to talk. Mr. John Ray Oronan, are you still there? Okay, we have Emar Ben raising his hand. Is this John Ray? Hello? Hello? Mr.
Mute na lang
John
ulit.
Ray.
EJ, can
Okay,
you
we have yeah, anybody who's raising also. Anyway, maybe we can have another question from our attendees. I think we have some students here. If students want to learn more about gene editing when they pursue their BS degree, which universities in the country they can apply to? Dr. Saportesa, please, and then Dr. Simora, will be the time that you can.
I think, I think we're both biased, Dr. Simora and I. It's only
Yeah.
the UP system. I'm sorry, we're UP. But of course, I cannot, there are other universities, because this technique is actually, what Dr. Simora said, is revolutionary. Most biologists are now into it, studying functional genomics, and studying what structure and function of different genes, regulation. It's really now one of the, I think, the technique in the toolbox of a molecular biologist is this one of the most powerful to use. I think it's up to you really. I
Mhm.
mean, you have to take the UPCAT.
Uh-huh.
Nag-ano na ulit yung UPCAT. So you really have to really work on it, study very well on the basics of the
Uh,
foundations
okay.
courses. So
Maybe you can talk about the strength of your university when it comes
yeah,
to.
you see, I belong to the Institute of Biological Sciences. So
Mhm.
this one, the biology degree, BS Biology in University of the Philippines, Los Baños, is already AUN certified, and it's one of the centers of excellence in biology in the country. So we have 5 majors. So I think this is kind of unique with UPLB, the BS
Mm,
Biology. We have major in genetics. We have
yeah.
major in genetics and cell and molecular biology. We have major in environmental science, major in animal biology, plant biology, and microbiology. So we have all those targeted specialities. We have a very good faculty. I think most, maybe around 65, 70% are already have their doctorates. And then we have laboratories, of course, and then we have also centers of research here at UPLB. We have the Institute of Plant Breeding, the Biotech, and of
Mm,
course, our next door neighbor is the International Rice Research Institute. So overall, I think UPLB can offer, if you're, especially if you're um, you would like to work on plants, since UPLB is also
mhm.
one of the premier universities for agriculture. So if you want to specialize in plants, I guess best bet is to really find, take an exam at UPCAT and try to
How about you,
register.
Dr.
Yeah.
Simora?
Yeah, come to UP. I've been telling everyone since when Isa invited me last time that my audience is high school students or high school students. I've been telling them to come to University of Philippines, Visayas College of Fisheries. We are also AUN certified like UPLB Institute of Biology. So
Yeah. Mhm.
we have a lot of we have programs for aquaculture, for marine sciences, for fish processing technology, for business management, fish business management, policy studies in fisheries. So come to UP.
Okay. We have one here raising his hand. Franz Mariel Garcia. Uh, you can shoot your question now. You can. Hi, Franz. Can we unmute Franz? Oh, anyways, I think we have exhausted our questions from or anybody. You can still ask our experts. If you want to raise your hand, and we will allow you to speak. I think there is no more. With that, uh, I think we need to end the open forum, the Q&A session, and we want to thank our two experts, Dr. Simora and Dr. Saportesa. I now give the virtual microphone to our moderator, Janine. Janine, you have now the virtual microphone. Thank you.
Thank you very much, Pastor Poppen, for moderating our Q&A session. We encourage everyone to please complete the evaluation form to receive your certificate of participation. Scan the QR code flashed on your screens or go to the link to access the evaluation form. This post-webinar survey will help us improve our future events and webinars. Again, to receive your certificates, you need to fill out the evaluation form. For everyone's information, we will be having the second part and third part of the webinar series in November and December this year. It will be focusing on gene drives and novel foods. If you're interested in joining the said webinar, please do follow us on our socials at isa.org. Stay tuned for our future announcements, and we hope to see you there. Before we wrap up today's webinar, we extend our sincere gratitude to our research speakers who shared their expertise and insights in gene editing. Thank you for accepting our invitation, and we are truly honored to have you as our speakers for this webinar. To formally close our webinar, here's a message from the Director of DA Biotech Program Office, Dr. Paul Limson.
Good morning, everyone. It is with great pleasure, although through this video recording, to give this closing remark. I want to extend my warmest congratulations to all the participants of today's webinar. While we may not be able to meet in person, the power of technology allows us to come together and discuss the future of biotechnology. I fully thank ISA for this opportunity. In recent years, our world has seen profound shifts, including a growing population, accelerated urbanization, and changing dietary habits among the public. These advancements, along with the pressing challenges posed by climate change, have exerted tremendous pressure on our agricultural systems to produce more food both efficiently and sustainably. Given this context, the demand for food has reached unprecedented levels, making it crucial for us to discover sustainable methods to satisfy this growing need. Today's discussions have truly underscored the monumental strides we are making in gene editing technologies and showcase the transformative potential they hold for agriculture and animal husbandry. From enhancing crop resistance to pests and diseases, to improving nutritional content and enabling more sustainable farming practices, it is evident that biotechnology serves a pivotal role in addressing some of the world's most pressing challenges. Gene editing stands at the forefront in the realm of biotechnology. Throughout our discussions today, we have seen how these techniques are not just theoretical, but are being applied to real-world challenges. We notice how gene editing in crops hold promise in developing varieties that are more nutritious, resilient to climate change, and resistant to pests and diseases. Imagine drought-resistant rice or wheat with enhanced nutritional profiles. These are just not ideas of the future, but tangible solutions becoming reality. Similarly, on animal biotechnology aspect, gene editing is paving the way for advancements such as disease-resistant livestock, improved meat quality, and more sustainable farming practices. From creating pigs that are resistant to viruses to enhancing the growth rates and feed efficiency of livestock. These innovations underscore the profound impact gene editing is having on agriculture and food production. I do hope and believe that our discussions today have not only highlighted the scientific and technological elements of gene editing, but also inspired a collective vision of how biotechnological innovation can drive sustainable and equitable advancements in the industry. With that, I would like to extend my heartfelt gratitude to our distinguished speakers, panelists, and participants for making today's webinar a remarkable success. Thank you, and we at the DA Biotech Program Office look forward to our next opportunity to explore these exciting advancements together. Mabuhay at patuloy nating suportahan ang Pinoy Biotech. Good morning.
Once again, thank you very much for joining us today. We hope to see you on the next part of this webinar series. Thank you, everyone, and have a great lunch.
Can I plug? Hi, Nawala.
Yes, Ma'am Ola.
I'll just plug. So as I said a while ago, there is the international virtual workshop, international, that is ongoing. And all of the presentations and videos will be posted also at the ISA website. There are a few questions that were not answered, especially on CRISPR or gene editing of livestock. We have gene editing of poultry, and also the small ruminants like goats and sheep. And
Yeah.
there are going to be some of those presentations were presented this morning at the virtual workshop. So when it's already ready, we will let you know. So maybe it'll be ready in 2 weeks' time. Thank you and have a great day.
Mhm. Ang dami ng ano yun.
Madaming tanong, no? Ang dami nag-attend. Very happy naman.
My last count is 270.
6. Yeah. Including us?
No, it's only the participants.
Sila lang? Wow.
Yeah.
Oo. Ang galing ni Janine. Thank you, Janine.
Thank you, Janine.
Thank you, Dr. Mabel. Thank you, Dr. Simora. Ayan na, ni-stop na ako.
Welcome. Ma'am, I will invite Isa dun sa lecture.
Opo. Yes.
Sure.
And if you can, kung gusto mo mag-invite ng mas maraming stakeholders like what we do, we can put it in the Biotech Updates newsletter.
Opo.
Kung
Sige
ito
po.
if it's, kailan niya siya?
December 16 and 17.